Hello, I'm Theodora, and I'm a student in Dr. Naj Barol's lab at Canada's Michael Smith Genome Sciences Center. Today I'll be introducing Meta-NanoSim, which is a metagenome simulator for nanopore reads. Metagenomic studies using long-read sequencing technologies, such as those developed by Oxford Nanopore Technologies, have been increasing in frequency due to the long read lengths it generates. However, compared to the short reads from Illumina, these long reads have high error rates and non-uniform error distributions, which necessitates the development of tools specific to long reads. Simulated data with known ground truth can facilitate the development of such tools. Shown here are a couple metagenome simulators that have been developed in the past few years, where the color of the arrows indicates a sequencing platform. As you can see, there's only one tool, MICROBIOME, that's capable of simulating ONT metagenome reads. However, it relies on NanoSim, an ONT read simulator developed by our lab to generate read profiles and simulate reads. Here, we introduce Meta-NanoSim, which, as I mentioned earlier, is an ONT metagenome simulator that's implemented in Python. Shown here is the workflow, which consists of two stages. First is a characterization stage. Here, ONT reads are analyzed to accurately model the read length distribution and errors in the metagenome. The output of this stage are read length and error models, which are then used in the second stage, simulation. Here, single or multi-sample datasets of complex microbial communities with specified abundance levels can be simulated. In addition to the simulated reads, error profiles, which indicate where errors were introduced into the reads, are output. So now I'll explain a little bit more about the two stages. In the characterization stage, read length distributions and error models are determined from primary and compatible supplementary alignments. Compatible supplementary alignments are included in downstream analysis to account for reads that align to circular genomes or plasmids, as well as any chimeric reads that may be present. The plot shown here shows the length distribution of the unaligned regions of each read, and it illustrates the importance of considering primary and supplementary alignments to ensure accurate length profiles. When chimeras are not detected, you can see there's a peak which is not present when chimeras are detected, due to the inclusion of compatible supplementary alignments in the analysis. Using the information from the alignments, we then model read length using kernel density estimation. Several aspects of read length are modeled, such as the length of the aligned region of each read, the ratio of unaligned region to the aligned region of each read, as well as other aspects. To model errors, specifically the probability of each error type, we use a Markov chain as shown here on the right. We also model error and match lengths using mixture models. Based on the empirical profiles generated in the characterization stage, reads can be simulated from the reference metagenome. During simulation, the species or strain to be simulated is selected based on the provided abundance profile. Then, we generate the simulated read lengths based on the length distribution. Next, we determine the error positions and lengths that will be introduced based on error models. We then extract the sequence from the reference and mutate it accordingly, and then add unaligned sequences to the 5' and 3' ends, the length of which is determined from the read length distributions. Finally, we output simulated reads and error profiles. To compare the performance of Meta-NanoSim and MICROBIOME, we trained on a mock microbial community consisting of 8 bacterial species and 2 yeast species, distributed on a log scale. The plot here shows the distribution of the total read length, which is represented by the solid line, and the unaligned regions on the ends of each read, which is represented by the dashed line. In both distributions, the read lengths of the simulated dataset from Meta-NanoSim closely follows the empirical length distribution. In particular, the simulated total read length distribution directly overlaps the distribution in the empirical reads. For MICROBIOME, which is in red, you can see that the simulated read lengths are inflated in comparison to the empirical reads. Although MICROBIOME uses NanoSim to simulate reads, it's using an older version which is unable to detect circular or chimeric reads. Additionally, the profiles used as input for the simulation stage are hard-coded. This plot shows the cumulative probability of consecutive matches and errors in empirical and simulated reads. Both Meta-NanoSim and MICROBIOME mimic the mismatch and deletion events well when compared to the empirical dataset. However, Meta-NanoSim simulates insertion and match events better than MICROBIOME. This is likely due to the reasons mentioned previously to justify inaccurate read length distributions from MICROBIOME. When quantifying the ONT metagenome reads with salmon, we observed that the experimental species abundance deviates slightly from the expected values. Meta-NanoSim can mimic this behavior in simulation by adding some random variation when selecting the strain or species to simulate. Shown here is the variation, which is calculated by subtracting the expected abundance values from the abundance values measured by salmon. and this is plotted on a log scale. A two-sample KS test was used to compare the empirical and simulated distributions. We can see that the p-value is higher for Meta-NanoSim, indicating that it's more likely that the variation in simulated reads from Meta-NanoSim and variation in empirical reads come from the same distribution, whereas the difference between CAMISIM and the empirical distributions is marginally significant but still substantial. As I mentioned in the beginning, the motivation behind Meta-NanoSim is to provide simulated reads to facilitate the development of bioinformatics tools specific to ONT metagenome reads. To show an application of Meta-NanoSim, we've benchmarked the performance of MetaFly, a metagenome assembler. The log-distributed mock microbial community was used to train Meta-NanoSim. Then several datasets were simulated, each with different number of reads. Specifically, we simulated datasets with 1 million, 2 million, 4 million, and 10 million reads. Each of these datasets were assembled with MetaFly, and the assembly quality was evaluated using MetaQuest. Shown here are 4 plots, the top showing the read coverage, and the bottom 3 are the genome fraction percentage, NGA50, and the number of misassemblies for each assembled species, where the color of the bar indicates the dataset used for the assembly. The x-axis is ordered by abundance, so the species with the highest abundance is on the left. For the 2 species with the highest abundances, the genome fraction percentage is almost 100%, and we can see the NGA50 plateau. However, despite increasing the dataset size, we still weren't able to attain high enough coverage to assemble the remaining 8 species, which explains the low genome fraction percentage and NGA50. Likely we could obtain better assemblies if we increase the number of reads simulated. In conclusion, Meta-NanoSim can accurately characterize metagenomes consisting of circular and/or linear genomes, as well as chimeric reads. It can then simulate metagenomes with accurate read length distributions and error profiles at user-specified abundances. Thank you for your time. You can check out NanoSim at the GitHub link shown here, or you can scan the QR code.